10x, and I work in the assay development department. So I'm mainly focused on the R&D side of developing assays with a specific focus on immunology and repertoire profiling. So I'll start by giving you a bit of background about what 10x does, and then dive into some data that we've recently generated, specifically using our immune profiling workflow. So I don't think I don't have to introduce to anyone in this room that the complexity of biology is immense. And when we're thinking about cells and the DNA in the cells and subsequently the RNA, when you scale this into the proteins and the number of cells in the body, it really, really is a very complex problem we're trying to solve if we're trying to understand how all these cells interplay and what the implications of things going wrong is and how disease comes about. So I think it's fair to say that if we're really trying to get at what's going on, tools that we have available to us or have had available to us until now probably aren't sufficient for us to get at the intricacies and the detail that we need. So at 10x, we develop solutions, or we like to think we develop solutions to this problem. We have workflows that incorporate instruments, biochemistry solutions, analysis and visualization softwares. And so it really is an end-to-end solution that allows you to resolve the complexity that you're trying to get at. So to date, we have products for linked-read genome sequencing and single-cell assays, and later this year we will be launching a spatial transcriptomics solution. So a little bit of a history about 10x. We launched our first product in 2015, so we haven't been around for that long. And as mentioned, that was the linked-read genome sequencing product. Our first single-cell gene expression product hit the market in 2016, and our first real foray into immunology was in 2017 with our immune profiling solution, which initially launched with just TCR, single-cell TCR profiling with gene expression, and we more recently added B-cell and then primers specific for mouse as well. Last year was a really busy one for us, and we launched 6 products. So our single-cell DNA, our single-cell attack, the third version of our single-cell gene expression product, and then at the top there in the blue, these are features of our feature barcoding technology, which is a way, so a sort of site-seek type approach that allows you to probe cell surface antigens or proteins using antibodies or other reagents that contain a barcode tag. And I'll talk more about that later. And as I said, later this year, we're launching our spatial gene expression product. So over the last few years, the number of publications citing 10x has risen dramatically, and you see that a lot of these publications are in the immunology or immuno-oncology disciplines. And I think that what we're doing is really very relevant here because, as I'll talk about later, just having one piece of information about your cells I don't think is enough. And so being able to piece together lots of different things and bring that together really gives you insight on what's going on inside a tumor or what's going on with the T cells in your body. So one example of one of these papers that was published last year in Nature was this paper where they used a single-cell gene expression assay to look at the different T cell populations within a breast cancer tumor. And they were able to subset out the different T cells based on their gene signatures. And so in the TIS plot in the center here, you see all of the different T cell populations that were identified. And then here in the heat map on the right, the breakdown by the gene signatures that were used to classify these cells. And they were able to identify a subset of tissue-resident memory T cells that had a specific signature that demonstrated improved prognosis in triple-negative breast cancer. So just one example of the sort of power these technologies have. So I'm mainly going to talk today about our immune profiling solution. So we started developing this product because we recognized that traditional methods for immune profiling, which typically rely on bulk measurements, so bulk repertoire sequencing or bulk RNA sequencing, and I'm sure you're aware that this just gives you an average readout of what's going on in your cells. The other limitation with traditional methods is the ability to look at multiple things at once. So typically you look at just what the cell surface proteins are doing, or you'd look at the TCR or the gene expression. And one of the things that I think is really important when you're trying to profile what's going on in the immune system is to have a complete readout of more than one thing that's happening. So our immune profiling solution allows you to characterize the gene expression, the T or B cell clonotype, and the cell surface markers from the same single cells. The TCR or the IG sequences are full length and paired because of the nature of the assay. It's single cell, so you get the paired sequences. We have high cell throughput, and as I mentioned earlier, the products are coupled with analysis and visualization software. So understanding peptide MHC recognition by TCRs, I really believe is central to understanding what's going on in adaptive immunity. And so in the figure here, what you can see are T cells, TCRs interacting with the MHC class 1 and the peptide, or the MHC class 2. And to really understand this process, what we need to do is break down this complex, very large factor puzzle into a couple of different things. So we need to look at the peptide MHC space. So this is like what are cells presenting to T cells that elicit the response. And we also need to understand the receptor of the T cell diversity. And if we can start to understand this, we can start to potentially build predictive models that will allow us to, in going forward, develop therapies. So our technology that was launched last year that allows us to do this is our feature barcoding technology. So if you imagine that in a normal assay, you'd capture your RNA molecules and turn them into cDNA and then generate a sequencing library from that. With the feature barcoding technology, you're effectively doing the same thing, but capturing these tags that are attached to antibodies or dextramers in the case of the reagents Immudex provides. So we have compatible partners that provide these reagents. So BioLegend have a whole range of antibodies compatible with the assay I'm going to talk about, are the Total-seq-C antibodies that they conjugate on the oligo for you. And then you can run these antibodies as part of your assay. And if they're captured or if they're bound by the cell, this oligo sequence is incorporated into your library prep, and so then you can get a readout of what's been bound and what your cell is expressing on the cell surface. And in a similar way, Immudex, who make dextramers, so if you're not familiar with dextramers, there's a similar approach to tetramers. They have a dextran backbone with multiple peptide MHC molecules lined along the backbone. And they also conjugate oligos to this backbone, and so they can be incorporated into our assay as well. So today I'm going to talk you through some data where we use this technology to look at two PBMCs from two donors. So we looked at a CMV seropositive donor and a CMV seronegative donor. 7,000, 8,000 cells for each one. And for the same single cells, we characterized the gene expression profile. We looked at the cell surface protein expression. So we used a panel of 14 antibodies from BioLegend, and we used antibodies that we knew we'd pick things up in our PBMC population with those antibodies, so CD3, CD19, CD4, CD8. We also profiled the repertoire of the T cells, so we got the paired TCR alpha-beta sequences for the T cells within the PBMC population. And then to probe the antigen specificity, we used a panel of 12 dextramers. And we used dextramers with specificity for CMV, since we knew one of these donors was positive for CMV and one was negative. And we also used dextramers for EBV and flu, just to see if there was anything in there of interest. So when we start preparing for these to run these assays, one of the things that we spend a lot of time on is sample preparation. And we have a whole group at 10x dedicated to making sure that sample preparation is done well. And they put out protocols and things that we share with our customers. And single-cell assays really highlight that if you put garbage in, you get garbage out. So it's really important to get your sample prep right. For the immune profiling assay, you can start with any cell type that has some immune cells in there. So that could be dissociated tumor cells, PBMCs, or purified BOT cells, whatever's of interest to you. When we're doing dextramer binding assays, we often do a monocyte depletion step. We find that monocytes are quite sticky and they sometimes bind to the dextramers and give you a bit of a non-specific signal. So we stain our cells with our antibody panel, our dextramer panel. We include some flow antibodies, which are different clones to the feature barcoding antibodies, and dense cell markers. And then we sort the cells. So when we sort, we're looking to really pull out the dextramer positive, so the antigen binding CD8 positive cells. So there are T cells that are really interacting with the dextramers that we've put in. So we gate on foreign side scattered on the lymphocytes, then look at the CD4, CD8, take the CD8 positive population forward, and then gate on the dextramer positive cells as well. And we sort these straight into a solution that we take into the assay. And as I mentioned, our team, our sample preparation team, write protocols on this regularly, and if you are interested in getting a bit more information there, you can pick up these on our website. Okay, so the workflow here is the overall workflow for the assay, including the sorting steps. The data I'm going to show you, we actually took the unsorted cells and the sorted cells and ran them side by side, just to get us an idea about whether we need to sort to look at antigen specificity, or also provide insights into what else is going on in the population outside of those antigen-specific T cells. So if we're sorting, we would label the cells, we would sort them, follow the gating scheme that I just described, and then load the cells with the reagents and the gel beads into the 10x chip. So we run this through our microfluidic system, which partitions cells into droplets. And each droplet, so this is shown at the bottom here, each droplet then contains a gel bead. And then on the surface of the gel bead are oligos that contain a specific barcode. So also within the droplet, you have a single cell and the reagents for an RT reaction. So then when you go through RT, every single RNA molecule associated with that cell, and if we're doing the labeling, all of the tags from the antibodies or the dextramers, get this barcode incorporated into them in the RT step. So for the downstream, you can then computationally infer which transcripts and which tags came from which specific single cell. So once we've generated our first strand cDNA, we amplify it and then perform a cleanup step. And it's this cleanup step that allows us to get at those tags from the antibodies or dextramers. So when we do a spray-based bead cleanup, the larger DNA fragments are bound to the beads. And so when we elute that off the beads, we use that material to generate our gene expression libraries, and then also amplify it for the TCR and subsequent library construction. The supernatant, which you normally throw away, we actually keep and make libraries from that as well, because those small fragments of DNA that are contained in there, as I said, contain those tags. And then that's your readout for what was bound to the cell. So let's hit the data. So first of all, here are our two donors, our seropositive donor and our seronegative donor. And we took the data from the sorted cells and the unsorted cells, and we combined it to see sort of how similar those populations of cells were. And you can see that in both donors, the sorted cells in green sort of cluster in specific areas of the whole population, which is exactly what you'd expect to see. So then when we go on to classify these cells based on their gene expression profiles and looking at the antigens they've bound, we're able to see like very diverse populations across both samples, which is what we'd expect, and that's great. Because we used a limited antibody panel, we're not able to directly identify every single population of cells just based on the antibodies in our panel alone. So, for example, megakaryocytes, erythrocytes, dendritic cells, they're classified based on the gene expression. But then for other populations of cells, having the antibody information is really, really useful. So, for example, We use the cell surface protein binding to look at the different subsets of NK cells that we can see here. And also the different isoforms of CD45 that are on the surface of the cells we can pick up using the antibodies, and it's much harder to see that in the gene expression data. So back to the workflow, I just wanted to put this here to remind me to tell you that we're now just going to look at the sorted cells only, because to focus on the antigen specificity, we think that focusing on a smaller population of cells that are specifically enriched for these events gives you a better insight into what might be going on. So when we look at the sorted cells from each donor, and I'll start with the CMV seropositive donor, you see that in the gene expression profiles, you mostly see CD8 T cells, which is exactly what you'd expect. There are some NK cells and very small clusters of B cells and monocytes that have just come through in the sort not being 100% clean. Let's say a similar story in the protein expression data. The thing subsetting these out is the CD45 RORA. And I should mention, so here, these are clustered based on similarity of gene expression, and then here, the protein expression, these cells are clustered based on what they bind to, so whether that be the antibodies or the dextramers. And then when we look at the antigen binding specificity, and again, this is clustered in the same way as this plot, we can start to identify clusters of cells that have specific binding interactions with specific dextramers that we've stained the cells with. So this is our CMV seropositive donor. So as we would expect, we do see some specificity for 3 different CMV PMHDs. We also see a cluster of EBV-specific cells and a small cluster of flu-specific cells, sort of suggesting past infection. But what's interesting here is when we look at the clonotypes that are associated with the cells in this cluster, the top clonotype only actually has 12 cells associated with it. And so that tells you that this is likely a latent infection. It's not very actively responding to the patient's not very actively responding to CMV. So then when we look at the CMV seronegative donor, on the gene expression and the protein expression front, the story is very similar. What's notable here is that you see many more memory T cells than you do naive T cells, which sort of started to suggest that maybe there has been exposure in this patient that isn't CMV, but is something else. And when we look at the antigen binding specificity of these cells, we see this huge cluster of EBV-specific cells. And the top clonotype here has almost 500 cells associated with it, so suggesting that this is an active response in this patient. We also see one other small cluster of EBV cells, and again, flu. But most of the people we look at have some flu specificity. So to dig into this data a little bit more, we wanted to break it down by every single dextramer that we'd put into the mix, and then look at the total number of clonotypes and what we determine as specific binding events for those clonotypes. So in these plots, I'll walk you through this slide. So we have the CMV seropositive donor on the left and the seronegative donor on the right. And then these heat maps show binding for the sorted cell population or the unsorted cell population at the bottom. And here, as I said, each row is a different dextramer, and each column is a specific TCR. So for this TCR, in the first column, we see a count of 16 total clonotypes, and 16 of them bind specifically to this dextramer for CMV, indicated by the blue. So the blue is statistically significant binding. When we look at the seronegative donor, we see these red events, and these are events where the clonotype is significantly underrepresented in the population. So there's this clonotype there, it's expanded, but it's not significantly binding to this specific dextramer, nor any other dextramer in our panel. So this suggests there's some other exposure in this patient that to really get it, we need to expand our dextramer panel to figure out what it is. The other interesting thing here, and I think we likely knew this already, but it's just nice to see it, is that if we look at the unsorted cells, we don't really see any binding events And I think this is basically, if you're in an unsorted population, these events are so rare, it's kind of like looking for a needle in a haystack. And so you really need to sort the cells for dextramer positive cells, CD8 positive cells, to get your sort of concentrated antigen-specific cells, and then you can start probing exactly what these cells are binding to. So of course the acid test is like, how does it compare to traditional methods that people use to typically look at these kind of things? And so first of all, I'm going to talk you through the top panel on this slide. So we ran our assay and we took the same data and then analyzed it in a different way to look at these specific binding events. And so what you can see here, this is for the CMV positive donor. And for each of the 3 CMV dextramers where we saw binding in that Disney plot a couple of slides ago, we looked at the cells that were specifically binding to those dextramers. And so on the y-axis, you have the log counts of binding events to a specific dextramer, and on the x-axis, you have a negative control. And the dextramer negative control just contains a peptide that shouldn't be recognized by anything. It's sort of a scramble sequence. And so to call a cell a specific binder, we have a couple of criteria. The first one is that it has to show significant binding to a dextramer, specific dextramer over the negative control. So you want these dots to sort of be coming up here as far as they can. And then we also have an arbitrary absolute number cutoff for the number of binding events we want to see. So that we get rid of a lot of the false positives and just the noise in the background. And you can see we see very different numbers of cells for the different dextramers, which was also reflected in the Disney plot by the size of those clusters. When we compare this to flow cytometry, the numbers are fairly similar for the most part. We definitely see a bit more in this high binder in the feature barcoding counts, but generally across the board, the trend is very similar. And so this really reassured us that we felt confident in the technology and what we're seeing. So another question we wanted to ask of this data was, of these TCRs that we see that bind to a specific dextramer, so a specific PMHD combination, is there any relationship between them? So if we see lots of different TCRs binding, are they similar? Do they diverge from the same sequences? And so we ran data for the CMV seropositive donor for the top 2 clusters. So the large cluster we saw, and then the next cluster, through TCR-DIST, which was published in Nature a couple of years ago. And this is a similar algorithm that generates hierarchical clustering based on the similarity of the TCR nucleotide sequences. And so what you can see here is that in the first PMHD binding event, we see this group of TCRs at the bottom that share quite a lot of sequence homology. And highlighted in green are the sequences that are also identified in VDJDB, which is an online database of TCR antigen interactions. So this sort of, again, gave us confidence in that what we're seeing has been previously reported and validating our findings. The reason you only see it for TCR beta is that most of the data in VDJDB was generated from bulk repertoire sequencing data, so it doesn't have the pairing. We did, however, find one paired clonotype for the other CMV PMHD that we looked at. So I hope I convinced you that the immune profiling solution and feature barcoding technology really allows you to fully characterize single immune cells. I've talked you through some data where we've seen that the increased phenotypic specificity from combining gene and protein expression really allows you to get at those cell types you might not be able to do with either technology alone. Our technology also allows you to look at the adaptive immune response from previously unknown infections. So you can think you know about your patient, but unless you're really profiling what's in there, there's likely some other exposure that's been there, and whether that's significant to your study or not, you'd have to dig further to find out. And as I just showed you in the TCR-DIST data, we can identify novel TCR clonotypes, but also previously reported ones that give us the confidence that what we're seeing is true. And overall, this sort of leads to a better understanding of TCR specificity. So the data I showed has been put together in an application note and is on the website alongside a data set that you can download. So this study was really interesting to us, and we wanted to push it a bit further and see what else we could do. So we went big. We decided to try and profile as many T cells as we could. So we got 80 million CD8 positive T cells from total 80 million cells from 4 donors. And we used a panel of 50 dextramers, so 44 specific PMHD multimers and 6 negative controls. After sorting for the specific binding events, we got 200,000 single cells, and then based on that stringent antigen binding criteria that we put in place, we get data readout from about 63,000 cells. And we generated this data set in a week using M8 Chromium chips, and as far as we're aware, it's the largest public data set of its kind that has been generated. So to put it into context, how big this is, we scanned the literature and looked at what other people have done, and there's a couple of papers out there where they've looked at 8 to 10 antigens, and then they see just over 2,000 TCR antigen specificities. And the VDJ database contains about 2,000 paired TCR antigen specificities over 83 antigens. And so in this data set, we have over 14,000 TCR antigen specificities for 37 antigens. And so that's 7 times bigger than the current known landscape of what's out there for alpha-beta TCR antigen specificity. And 23 of the antigens that we had in our panel have no previously reported pairs in VDJDB. So this is completely novel TCR data. So we actually just released this data set on our website last week. So if you're interested in looking at lots of TCR antigen interactions, please go ahead and download it. And a colleague of mine will be giving a webinar on this in a month, discussing in-depth detail of what was done there. And that's it. I'll take it.